Good morning, everyone. Do you hear me well?
Yeah.
So I assume there was no questions, so everything was crystal clear. You have everything sink into your heads. Now you know almost everything, not everything about olfaction. But today we're going to talk about how to understand the sensory coding from behavioral perspective and in olfaction, basically. Again, please interrupt me. I hope that Vijay will follow the chat. For me, it's a little bit difficult to follow the chat, but if somebody will kind of alert me, that would be good. I'm happy to break and answer the questions. Have a break and answer the questions. So I would like to start with an interesting example, not an olfaction. Imagine we study the tennis player. We study the behavior, the trajectory and the behavior of the how the tennis players serve the ball. For full disclaimer, I'm a very poor tennis player, so please excuse me. But what you do, you have a fantastic camera that can record a very high precision the trajectory of the tennis player racket, okay? You record this and you can measure this trajectory, okay? You can measure this trajectory. But imagine just for a while that your camera doesn't see the ball. What the camera see, where the ball landed either here or here. And my question for you, can we predict position of the ball given the trajectory of the racket? Oh, of course, sure. You see the trajectory of the racket, you see the ball, you can predict. But the most interesting part, that the strongest prediction of the position of the ball will be where the racket ends, here. This position strongly correlated with the position of the ball. So you record many tennis serves, you find where the ball landed, and you write a paper. The final position of the racket is representative of the position of the ball, in the other side of the tennis field. And fantastic discovery, you find the code. Well, obviously, there's a problem here. Because after the ball left the racket, that trajectory has no causal effect on the position of the ball in the field. I hope that is clear. So what's interesting here, that the position of the ball in the field, these two spots are defined by very small deviations before the ball left the racket. So that part of the trajectory is important. Now, you don't see the ball. How we understand this? What our way to make a causal relationship between the trajectory of the racket and the ball? Well, there's multiple ways. First of all, we can study the physics. And we can study the physics and see where the ball left, and eventually we come you study the ballistic movement, and eventually you make your model more and more accurate. You take into account the rotation of the ball, you take into account the friction of the air, blah, 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 and you find, ball left the racket here, but not here. Okay, good. But there's other ways to do it. We can actually perturb. We can perturb and we can say, so if you perturb the racket here, definitely ball will fly somewhere else. If you perturb the racket here, nothing happens. So that's basically, I want you to carry in your mind that finding good correlation in the neuron code about some representation, even either of the stimulus or the behavior, does not mean that has any causal relation, and actually they're related to that. What's happening here, that position, just to be very clear, is a consequences of the small movement here, and the position of the ball causally related, but these two not causally related. So, I mean, it's important to study all this stuff. It's important to get an intuition. but we do need to understand how to deal with the neural code. And in general, our approach always was the same. For many, many years, we studied stimulus representation. We presented stimulus and we measured neural representation. And more and more now in neuroscience, we start caring about the behavior. This function has been studied a lot. We know a lot of math. We know a lot of, how code adapt, information transmission, linear, nonlinear modeling, approximately 50 years of work. But what we actually now interested is how neuronal code evoke behavior and how to actually mine, how to think about this error. And what can we think about the code by studying this relationship? And that's what I'm going to talk. And then I come back and show how that actually relates to our understanding of the order coding. Basically, this is my overall general philosophy. And to study this, we'll use perturbation. But we'll see how far we go. So let me briefly remind you, so you didn't have any questions, I assume you remember everything, but nevertheless, let me quickly go through what about organization of olfactory system. Odor gets to the nose through the inhalation. This is important. They reach epithelium. From there, information goes to the olfactory bulb and to the rest of the brain. There's other molecules interact with the receptors, receptor neurons that are located at the cilia of olfactory receptor neurons. There are multiple different receptors. We have a whole variety that sense different features of the odorants. It's kind of very schematic view of that. And mouse have 1,200 receptor genes. Humans have about 330 receptor genes. And when the receptors interact with the ligand, they activate it, they depolarize the cell, and the signals goes to the rest of the brain. Where does it go? It goes from the olfactory epithelium through the olfactory nerve to the glomeruli. What important, very, very important here, that the cell expresses one and only one receptor gene, and all the cells that express the same receptor gene converge to one glomeruli, which is, it's not, all receptor converge one or two glomeruli, so one glomeruli have only accents of the same receptor genes, but we have a few glomeruli like that. At least this is a dogma. To be careful, it's never been seriously contradicted, but I mean, while that many receptor converge to one glomeruli have been proved, you cannot just take it a little bit cautiously when you think about it. It's never been seriously never been either proved or disproved. And from the glomeruli, signals goes to the mitral cells. We talked a little bit that is a huge amplifier, that integrator and amplifier. So it integrates the signal from the approximately 5,000 receptor cells and sends to approximately 20 mitral and tufted cells. And different orders, and then it goes to the cortex. And different orders excite different patterns of receptor and glomerular activation that can be very nicely studied with imaging. Again, have in mind that when we study imaging, we unfortunately have access only to the dorsal part of the glomerular layer. And there are attempts to do much more recording. This is approximately 10-15% of the all glomeruli, but that's definitely beyond. So some of the work we're trying to, for example, just to inspire your imagination, we tried to develop the methods for ultrasound recording of all glomeruli simultaneously, but that's still in the future. And different glomeruli, different orders evoke different pattern of glomerular activity. And we can study this. Now, what we talked yesterday, we talked about electrophysiology, but there is another method to do mitral cell decoding. Mitral cells located, the dorsal layer of mitral cells, approximately 300-500 micron below the surface of the brain. and we can study them with a two-photon microscopy. And this is a beautiful picture of the mitral cells taken by with two-photon microscopy. And actually, current activity indicator became faster and faster, and we actually can start measuring. Sorry, I actually will show you the movies. Hold a second. Is the movie running? Oh no. Oi. My bad. It's actually the movie, and okay. You need to believe me. Sorry, it didn't run on my laptop. Oh, iPad. So you can study the temporal pattern of activity of mitral cells activation, and you can study this and try to understand the code. So what do we have? We have the following system. We have stimulus that are represented on the level of the receptor or glomeruli. They are represented on the level, then they transform to the mitral cell level, and then behavior. What do I mean by behavior? It's infinite amount of different behaviors. We can think about it in different way. Oh, by the way, here the movie is working. So if you're watching here, here's the movie, and you can see how different mitral cells get activated, becoming greener, and then disappear. Sorry for distraction. So behavior, we can do many different behaviors. And the very simple is you present an order and mouse need to lick and detect the order, or lick left, lick right, do something. So basically, I just call it behavior, but we can discuss in more details what that means. Basically, we need a full conscious response to an order, let's say detection. So we can study the code on different level and try to understand what feature of this code are related to this or that animal behavior. But, and this is the classical schema that I described, stimulus, neuronal representation one level, neuronal representation another level, behavior. What I talked to you that we need to be very careful of finding the deceiving neural correlates either with stimulus or with behavior, which may not relate actually processing of the information of the system. So to tackle this problem and try to understand this on different level, we actually do stimulation. How can we do the stimulation? And I give a very brief introduction about the methods, and then we'll go to the think about the actually studying the code with this, some of these methods. So we can put activity in the or cell activator or options into receptor neurons, into glomeruli, and use a projector. It's actually a relatively simple technique. You can start shining patterns like that, activate individual glomeruli. And this is one technique when you can stimulate individual glomeruli. We need to calibrate that you do stimulate individual glomeruli, that you kind of localize the stimulus, and on and on and on, but that's quite achievable. What can we do on the next level? Next level, mitral cells are located deep in the brain, and normal light doesn't penetrate so deep. But there is now techniques that is developing in a few labs, and we are we try to develop a technique to use so-called two-photon computer-generated holography. How it works? Basically, the center of this is the spatial light modulator. When you change the pattern of the change the phases of the coherent light and create a very specific focus on a specific target inside of the brain or in 3D. But differently from the lens, where you can do that focus with single lens, you can actually do simultaneously multiple patterns. And you generate holographic pattern. You shine the two photon bright light. You need two photon. So because you want to activate only this portion of the tissue, but not other. and you can simulate. You kind of embed the cells in the ball of lights, and you can start playing on neurons like on a piano. So something like this, and you can simulate individual neurons, and we can calibrate individual simulation, and on and on and on. Now we have two. I need to tell you, this is technically still very, very difficult. 20 years ago, few labs have a two-photon microscope, and you do need to be almost optical physicist to operate it. Now you can buy turnkey solution, and you don't need to know much. But currently, it is still on the level that you need to have a lot of technical knowledge to do this stuff. I hope in 10 years it will be or maybe faster. So what do we have? We have again the same system that we can perturb on this level, and we can perturb on that level, and we can study from the stimulus to the behavior. Okay, so what question can we answer? The first question that comes to our mind is basically probably inspired by the movie The Matrix. I don't know how you're familiar with this movie. It's one of my favorite ones. By the way, maybe at the end of December, there's a fourth Matrix coming out. I have no idea. I have low expectation, but still hope that it will be good. But basically, can we emulate the object? Can we emulate the object inside of the mouse brain by stimulating the patterns of activity on any level without presenting the stimulus? The difficulty with that, how you get a report, because if you do it with humans, you can ask the humans, do you smell a rose? Or what's going on, basically. But how you ask animal to report that you actually create an other object in the mouse head. And I'm happy to discuss it maybe at the end of the lecture. We're actually working towards this pattern, but it's still far away, and we still cannot emulate the perceptual object. But if you think about it, my belief that among other sensory systems, maybe olfaction will be one of the first where we can do this. We'll see. Second thing, so the second question, what feature of neuronal activity are relevant for specific behaviors? And we will discuss it in details. Because I give you an example with concentration before, and there's many features that are correlated with specific features of a stimulus. But what I use to extract useful behavior, we don't know. And we can try to answer these questions. And let's say the most basic question here, what feature of neuronal activity are perceptually accessible? It may sound kind of a little bit, superficial. I'm not sure that, we need to think through these specific questions, and I'll give you an example and we'll discuss it in details. But if you have questions now about the questions, please raise your hand. Something in the chat? Okay, no, okay. Okay, so as I said, studying two-photon holography is still very difficult, and we actually start doing this. But to get an idea of a conceptual approach, we first work on the first level, on the granular level, and try to answer some questions and try to understand what can be done here. I'm much more interested now in the conceptual approach, but because if time, we'll have enough time and we'll have some questions, I can talk about what can be done on the next level, because at the end, you want to stimulate any neurons in the brain and evoke some memories, create the pattern, and on and on and on. But let's just approach it with the simplest case. So that question is we cannot answer. Can we emulate perceptual object yet? And let's focus first on what feature of neuronal code are perceptually accessible, and I wanted to elaborate on it. You remember this example? This is from paper 2006. It's quite 15 years old paper, but still very, very convenient to give this example. If you increase concentration, you evoke more and more glomerular responses. But if you look at the specific glomeruli, glomerular 1 and 2, you can see that amplitude is going up. You see, as you increase concentration, amplitude is going up. So maybe you can, can animals sense a different number of glomerulus? Probably yes, but what sensitivity of this method? Can animals sense the individual glomerular amplitude? Or can animals sense the timing? So we can, when you present an order and you change concentration, all these parameters are changed simultaneously together, and you don't know what features are actually extracted during the real behavior. So how can we check this questions independently? If we have a full control of the order code, we can discuss the different mode of the order coding scheme, order coding. So the first code, when you just need to count of number of glomeruli, you don't care about the amplitude. It's a basically binary code. And it's a binary code, and you just need to count the number of the red spots. The second code carries more information because each glomeruli has analog response, and you not only count the number of glomeruli, but you also measure their amplitude. And the third one is actually timing, because different glomeruli activated at different time. And when I said timing, you have internal clock always. It's always locked to inhalation. And just let's use it as internal time axis in the system. How you check it? So to check the first mode, you basically need to make any mode to discriminate one pattern versus another with difference of one glomerulus. To check the second mode, you just need to be sure that animal can discriminate different amplitude of the glomerular activation. And to check the third mode, you need to check that animal can discriminate different timing of glomerular activation, let's say in the sniff cycle. Now, with orders, as I said, you cannot do it because you cannot individually manipulate glomeruli, but with applications, you can. And that's exactly what has been done. But for this case, we used the mouse that has a single glomerulus with channel rhodopsin, and we basically stimulated with a diet laser. Basically, we activated an individual single glomerulus with channel rhodopsin, and we trained the mouse, we recorded a sniff pattern, we presented an order, we trained the mouse to respond, to detect the stimulation, over the single glomeruli. After mouse has been trained for order detection, this is result. We presented the laser pulse, performance dropped, but then very quickly recovered, okay? So that is, so mouse can detect single glomerulus. And they were completely, when we simulated some portion of the pulp that is free of the channel rhodopsin axons, the performance of the mouse was on the level of chance. Okay, that's good, that's a good result. Now we need to measure at which level do they stimulate. So we train the mouse on the go, no-go task, so mouse leak versus not leak of the watery spout, when we increase intensity of the single glomerulus activation. And on the very lowest intensity, all mice perform on the chance level, and then the above approximately 3 milliwatt, they saturate. So important part, we need to think carefully about all these parameters and behavior, because it's a whole science how to do proper behavior. I don't have time to discuss it all, but please pay attention, because that sets you up how to do behaviors, how to interpret results, and how to do different controls. So we choose that intensity pattern. So it is not, so this, when you blast the light too high, the performance can be saturated, but in average, all the mouse performs, so average performance was 75%. So we choose as intensity pattern about 1 milliwatt that was detectable but not saturated. This is important. So first we train the mouse on the task, no stimulation versus stimulation, and all mice performed about 75%. That's the result that I showed before. Then we do it with the others. We stimulate other glomeruli, and we edit one glomeruli. That's exactly the experiment what we discussed before. And all the mice for different orders performed exactly the same way, as if there was no order. Their performance was almost the same. Well, maybe you blast the light so much so you almost burn it, and other animal doesn't pay any attention to the odor. So we did another experiment. Know that all these odors, hormone, hexanone, and lactonic acid, do not excite the glomeruli of interest. Now we choose another odors that excite the glomeruli of interest. So if you have a very strong stimulation, mouse would detect it anyway. But if the stimulation is comparable with the other stimulation by these odorants, by these ligands, they would not detect it. And that's exactly what happened. To prove, this is a control experiment, but very important, that when an odor activates these glomeruli, additional stimulation does not lead to detection, while exactly the same amplitude of stimulation lead to detection. So in this case, we did prove that animal can detect single glomerular stimulation in a normal range of stimulation. The next task is to do it to check the amplitude. So we train the mouse to discriminate 20 versus 1. Note that we need always to present something that it's not a detection task. We're not doing 20 versus 0, because maybe 1 milliwatt it's not detectable, so it will be detection task. It's a discrimination task because 1 milliwatt is still detectable. 20 versus 1, 20 versus 5, and 20 versus 10 milliwatt. And animal detect everything, and when the ratio between stimulation became very low, some animals fail to detect. We can discuss about actual range and the actual level of detection, but we established the range of detectability. In the timing case, we stimulated sometime after the onset of inhalation. We measured the odor, the pattern of inhalation. So this mouse exhaling, then inhaling, pause, exhaling, inhaling. We measured with a pressure sensor in the nose, and we stimulated exactly 25 milliseconds. And mouse need to respond go, and sometimes later we stimulate it with 100 milliseconds later, and that is no-go stimulus. And we change this parameter and see when mouse failed to detect. And mouse were able to detect everything until 25 milliseconds, and 50 milliseconds failed to detect. We can go on and on and on. I'm showing the type of the experiment. I don't want to go into much more details. We can think how the signals is interpreted, what that evokes spikes, and how this affect code, and on and on and on. But the bottom line, that all three modes of information transmission, binary, amplitude-wise, and temporal, are perceptible. Animal has ability to sense all this feature by individually. So basically, in this case, concentration can be detected in all ways. And now we can do a lot of different stuff. We can actually manipulate, compare absolute values, and no. Let me give you another example. So people study in vision how you detect the distance to an object. And the real world provides gazillions of cues. If you look at the distance perception, you find approximately 30 different cues that humans can extract from the scene. taking, you can binocular vision, context, really kind of the focusing of the eye to one object to another object. No. I'm not specialist in this field. Look it up, and you can see, for example, if you close one eye, you can still try to understand which arm is or which objects are further away, which is closer. You can do it on the you can judge, for example, if you look at me not on the slide, just by context and on and on. So you're extracting gazillions of different bits of information, trying to judge the relative position of the objects. And we can envision, we can manipulate them individually and try to build how different features are combined, how different features are affecting, how important is binocularity, for example, no. But we do need to know which is perceptible, which is not. For example, if you have one eye, there is no binocular cues, period. And on and on and on. So this is kind of establishing the background. But how can we study in the code, not on the stimulus field, but in the code that different importance of different features? And this is a difficult problem. And related to intensity, specifically how animals make a discrimination of concentration or judge intensity, we didn't have much progress, even though we know how to approach this problem. And please, I'm always open for suggestion, and we have people interested. I am happy to discuss it. But we actually try to develop a general approach of studying the relevance of different perceptual features in the code. We developed I think, a new paradigm that allows us to do this. And what features of neuronal code are perceptually accessible? Are perceptually accessible we discussed, and what features of neuronal code are relevant for specific behavior? This is the topic of the next story. Do you have any questions about the previous story? I actually try to, guys, provoke you. Please ask the questions. Just give me some feedback. It will show I'm very much for the interaction, because I'm moving forward, but I don't know if the previous kind of ideas have been perceptible, have been perceived, detected, and on and on and on. So please raise your hands and does the distance of the smell source affect the amplitude? Absolutely. But you probably have the whole lecture about order navigation. So how animals navigate, this is a complicated behavioral task, and what feature from the stimulus is extracted is super, super important. But it definitely does not solve the problem just by measuring the amplitude, even though it can measure the amplitude. Okay, let's move, let's ask the next question. What feature of neuronal code are relevant for specific behaviors? We decided to do the following paradigm. It's some thought-provoking. We decided to abandon orders at all. Orders are, nasty. It's difficult to deliver them. It's difficult to control it. So let's do a pattern. Just a pattern that looks similar to the orders. And we stimulated a bunch of spots on the olfactory bulb, A, B, C, D. And we trained the animal to discriminate this pattern versus another pattern, which is called non-target. When animal get target pattern, go left. When animal get non-target pattern, go right. But what we did, we basically presented not a single non-target, but many different non-target, completely randomly. The only thing that we kept is the number of spots and the temporal window. So non-target never repeats. So whenever animal sense the target pattern and go to the left, any other to the right. This is analogous of you asking if it's an apple. So you can ask the question, oh, is it a coffee? Oh, this is a coffee, but coffee versus whole other world. Tea, apple, banana, rose, no. So if you get your coffee, you're happy, you go to the left. If anything but coffee, you go to the right. So an animal can do it. Now, when animal can perform the task, on a well-trained animal, we can modify our coffee. We can modify our smell in a very controllable fashion. We basically can substitute one glomerulus, or we can change the timing of the one glomerulus. And we can ask how the detection of the pattern fails. Are you still perceiving the same apple? Are you still smelling the same apple? Or this is something different? And the logic is the following. So if if the performance at the target level is approximately 95%, it goes to the left. If you present non-target 5% and making mistakes, animal almost always make mistakes. So you start doing perturbation, different perturbation of the code, and the performances are dropping. And the level of the drop of the performance tells you how much it drops. Let's do different color. How much it drops tell you how important this perturbation is. So, for example, if it doesn't change the target recognition, this feature is not important. Maybe it's not important. Maybe this pattern will be exactly the same if I do this. Maybe animal perform the same apple if I do it all at the same time. We don't know it a priori. We don't know what features are important, what features are not important. But if the perturbation significantly decrease performance, that is very important feature. And the level of relevance can be judged by the drop of the performance. Hope this is clear, the logic. It's kind of, again, analogous. Imagine you hear the song. And now I start playing the song slightly differently, and some minor manipulation of the notes still allows you to recognize the song. I wouldn't dare singing on Zoom, but dramatic manipulation that immediately drop the performance. You would never recognize it if I significantly change it. And I want to quantify this. Okay, we train the animal for 6 patterns in approximately 200 milliseconds, 250 milliseconds. How you quantify performance in these experiments? Animal going left and right, and when I present the target, it needs to lick on the left port. And if I present the perturbing the third pattern, it making mistake. If it goes to the left, I assume it's performing as like a pattern, and this is exactly what's happening. So this is going to the left port, statistically. I measure the number of times they're going to the left port, and I estimate the probability for a given perturbation. And if the performance here, that I know that performance dropped 45%. How much latency can exist between activation of different targets in a pattern for which mice can learn association between targets in a target pattern? How much latency can exist between activation of different targets? Hold a second. By target, I call the whole pattern. This is a target pattern. You think how much latency between individual spots, or I'm not sure. Maybe you can unmute yourself.
Yeah. Let me try.
Okay.
I'll just unmute.
Shriram. Shivas.
Shivas, can you go ahead?
Hi, can you hear me?
Yeah.
Yeah, I did mean how much latency can exist between the activation of spot A and spot B or spot B and C and so on. So basically how much, like what is some time interval where the mouse can form an association for the complete pattern?
So very fair question. And what we did, we make this pattern more or less similar to the pattern of the other activation. The only thing that we didn't do is that we did not, so we choose some special pattern and we didn't match it to the other. Individual spot, and I didn't have time to talk about it, actually we put electrode inside of the olfactory bulb and measure mitral cell response, and we check that individual spots evoke very similar response like another, and we more or less activate the similar number of glomeruli statistically, but we never match the patterns. This is a very hard problem to match the pattern to the other, but it's statistically roughly the number of the spots and number of the temporal window is the same like in the order, but we didn't match precisely statistics.
Right, but over here, it looks like the activations of different spots were kind of overlap. But say there was a time interval between of like say 100 milliseconds between the activations of different spots. My question is how much of a time interval can exist for mice to actually form an association between, because over here, the target pattern is A first, then B, then C, and so on, and the non-targets are kind of more random. So say if there was, okay, so this is 80 milliseconds, but is it also overlap? Because it looks like it's overlapped here. So
it is overlapping, and it is actually, in some sense, arbitrary choice. We need to start with something.
Right.
And we can argue about it. So first of all, and that's a super important question, how similar it is to the order. And to tell you the truth, we don't know. But what we can say that when we train the animal, all animals learn this discrimination in approximately from 1 to 20 sessions, which is in the bottom kind of ballpark similar to the other performance. You see what I'm saying? So it is not completely outside of the range of animal performance. This is super important. We discuss this a lot because, you know, what we're actually doing, what is this pattern for the animal? It may stink, it may be very pleasant, we have no idea about animal perception, but all the behavior was similar to as if it is an order. And mitral cells activity was similar to that evoked by the others. Beyond that, I cannot say, and it's a very fair critique, but let's see the result and then see. It's not a proof of other code, is the hypothesis, is kind of the way to study this, and we'll see how it is, how to connect it to the real code, okay?
Right, thank you.
Yeah. Do individual order molecules activate a similar number of glomeruli? More or less, you know, it's also a difficult question because individual orders can activate from 1 to a few hundreds glomeruli, and all of them are detectable. So this pattern definitely fall in the range of the natural orders, because natural orders can be detected from, as I said, 1 glomerulus to hundreds. This is something in between, so that's not a problem. So we first train the mouse to discriminate target versus one non-target, and then edit multiple non-target, and all mice perform the task. It actually was, as soon as we get this happening, that was a big success. We can now start manipulating. Now, have in mind that mouse always train on this task. Mouse is trained to do A versus B, C, D, X. And it always reinforced for this task. And on a small number of probe trials, we perturb the pattern. And there's some behavioral tricks that we can do. For example, if you do perturbation, you need to know, do you reward it or not? If you do reward them, then mouse learn this. And you can say, well, maybe it's a learned association. Now, if you don't reward, so how to deal with that? So basically what we do, we actually reward only probabilistically. So when mouse making a correct choice target, we reward, let's say, 75% of the water. So if mouse make mistake, it doesn't know if it's a probabilistic not getting water or it's an error, and it continued doing this. So this is called partial reinforcement. We implemented partial reinforcement, and we start doing probe trials. This is super important in behavioral science, in behavioral experiments. The first thing, we perturb, we swap a spot for a different spot. On the spot, multiple spots for a different spot, randomly. 3 random spots, and we see the performance, how the performance degrade with the number of spots. And you can see that if 0 spot perturb, the mouse performance is, well, this is a target pattern, and it's almost linearly decays as you increase the number of spots. Now for one, now this is the number of randomly spots, but spots are different. So the next experiment that we did, we basically, we perturb only one spot, but we measure it as a position of the spot. First, second, third, and on and on and on. And what we discovered, that the perturbance of the first spot was mostly the most important. The drop of the performance was the highest, and when we perform the 6th spot, it almost doesn't change performance on average. So you see the general trend. And that what we called so-called primacy effect. Hold on a second. We, sorry. Just a second. And we feed the linear regression to multiple spots, because even when we perform multiple spots, we measure the importance of individual sport for the general performance with a logistic regression. And we basically feed the data for individual sport and multiple spots simultaneously. What was important, that we tried to add here a nonlinear term. So can the performance be fully explained by individual sports, or we can add additional terms to explain 2-spot stimulation is not additive. And without going into much details, we found that that term is not relevant. So the perturbance of individual spots are linearly additive, and we have so-called primacy effect, that the first spots are more important, and I will denote it by the blue part here. The initial spots are much more important for detection of the target. And that's what we call primacy effect. Okay, we establish the spot shift. Now what about timing? We can shift the timing of the first glomeruli, and as soon as we move it to the late in the sniff cycle, the performance dropped here. Second glomerulus, second spot, and all other spot. And what you see, sorry, oops, just a second, try to. What you see that the performance on the later glomerulus almost doesn't change. The performance on the later spot much less perturbing behavior than earlier spot. But what we notice also that, for example, if you look at this, that performance and this performance look differently. So when you move spot forward, fifth spot forward, the performance go much, drop much faster than you move it back. So we independently feed positive and negative shifts, and this effect can be explained again by the primacy. Basically, the earlier part of the sniff is more important than the later, and when you shift the spot to the earlier part, you're affecting the behavior stronger than if you shift it back. And that is kind of the most profound probably here. Now we do again linear regression, and now we see the effect of multiple spots, shift of multiple spots simultaneously, and we shift whole the pattern. If we apply the linear regression questions to the previously detected from individual spots, the performance dropped much faster than was in experiment. The fit, the model was much faster. So something is going on different. So our idea was the following, that the animal is much more sensitive to perturbation of relative timing versus perturbation of the global pattern relatively to the sniff. So what we establish, we measure the shift, we measure the shift to the central mass, the central mass, and we measure the shift of the individual glomerular relative to central mass. And we basically did more complicated regression and found effect of the central mass. And then the performance was very well described by the shift of the central mass with independent variable and the shift of individual spots.
Dima, there's a question in the chat.
Oh yeah, okay. Would adding a delay period in the task increase the dependence of later spots? Delay period in the task, you meaning between trial? Sorry, I can again, Sharam, can you unmute yourself and let's talk, because this may be important and I'm not sure I understood the question.
Hi, can you hear me?
Yep.
Yeah, so I was asking if the task included delay after the presentation of the optogenetic stimulus, after which the reward was given.
Absolutely. Super cool question. Thank you very much. Because what you can imagine that animal is impatient to wait to the end of the task, or the pattern, start leaking. We actually did a lot of controls. I just didn't have a time. So we actually delayed for significant amount of time. Animal cannot leak until 300 milliseconds after the end of the task. And it doesn't affect the performance. But you're right in some sense that as information going in, and if you measure actual leaking, if you don't put this restriction, occasionally animal making decision earlier. But that is okay. The early decision is so they get the first information and they already know this pattern is more important. Okay? So it's basically putting reward much later does not change the performance, but animal does extract more information at the beginning of the sniff than later the sniff. Did I answer your question? I assume.
Thanks.
Yeah. Okay. Yeah. So, and we kind of build a model of this processing because kind of we try to understand how this behaviors actually can be modeled and processed, because modeling allows us to extract the neural correlates of this process. Basically, if you have two patterns, red pattern is target and blue pattern is perturbation. We first convolve it with each individual channel. We convolve with the kernel with a characteristic tau activation, and the amplitude of the kernel decay with the tau primacy. So this is a very important part. So if the spot activated early in this new cycle, its kernel has large amplitude. If later, it has a small amplitude, and the amplitude of the kernel decays with the tau primacy. And we created the same pattern for two different. We convolve for all individual channels for target and non-target pattern, and we estimated the position of the central mass, align them relative to central mass, and measure the difference. And that is the measure of the S, the area under the curve, between the curves, the differences, is the measure of the pattern differences with some weighting coefficients. And we also added the term for the time shift. We penalized for the time shift, added that summate to both terms and measure perceptual distances. So what we have here, We have the model with relatively small number of parameters. It is tau activation, tau primacy, tau center of mass, and 2 weighting coefficients. And we tried to fit all our data, and we were quite successful. And what's important here, this is so-called spatio-temporal template matching model. People who study vision often apply spatial matching in vision. So you apply some pattern until how well it matches in the spatial dimension. In audition, people use temporal template matching. How well temporal template matching, temporal pattern is fit to the one to another, and this is called temporal template matching. In our function, we propose spatial temporal template matching, where temporal and spatial components are important, and we can measure the components' importance of individual factors here, and we basically feed the model, and the rest is in some sort of technicalities, because we were able quite well to feed the model with all these parameters, and even did some predictions. Again, I don't want to go to much details for the pattern that we never presented. In conclusion, okay, I do have some time still. Early spots are more relevant than later. This is called primacy effect. Spatial channels are independent. Each channels are independent and they're additive. Strong sensitivity to relative timing and weak sensitivity to global shift of the sniff cycle. And we have spatial template model for perceptual distances. Do you have a question about the series of experiments? Because I would like to kind of discuss a little bit the significance and interpretation of these results. How the capacity of the code affected by the primacy model? Great question, and let's discuss it in application to the others. And that will be exactly my second point, and we'll talk about it specifically and try to talk about that effect. Very important question. So first of all, I mean, this was not an order, so that was a pattern, but animal performed some natural behaviors, and reported back significance of these features of the code. Now the question, can we actually test it with the orders? How this is relevant for order perception? Is it has something to do with order perception, or this is artificial system? The next topic that I wanted to discuss it, when you present a real order, it does stay the same. How can we check it? Basically, look, if you measure any orders, you can image them and you can parameterize the code here, but other rules are the same when we do it with pattern simulation. And I would like now to shift the gears and give you some experimental approaches and some ideas why primacy coding is very, very relevant to order perception. And it actually comes from the basic kind of understanding that recognition of the order should have some invariance. When you recognize a face, you do recognize the face independently of lighting, its position on the screen, its angle, and on and on and on and on. So all objects in the world have a property of recognition of the objects on the world has a property of invariance. And the same thing happening to the orders. And that hasn't been really studied seriously. And this is super, super important for understanding the code. But again, that should be thought through the lens of behavior, because invariance is not something that you can understand by studying this relationship, how stimulus relate to this pattern, because you need to know what is important and not important for the animal. And one of super relevant feature is order recognition independently on concentration. So concentration invariant order recognition is one of the basic features that let's try to capitalize and see how that actually leads us to some ideas what we mentioned before. You recognize coffee independently how far it is. You may have different judgment, and you can extract much more notes from this coffee than from a Starbucks from far away. Coffee, somebody would, well, it's still considered to be coffee. But what create the object coffee independently of its intensity? And let's go back to the basic biophysics. Let's start thinking about the origin of the code and how this may be relevant and connected to the primacy coding. Let's go to really basic stuff, receptor-ligand interaction. So when you increase concentration, the activity of receptors can be defined by the sigmoid function, which is with the critical part is the threshold when the receptor activated. What is interesting here that receptor is the dynamical range of the receptor is much more narrow than the dynamical range of the whole system. That is a biophysics. This is basically the basic property of the receptors. But for the same ligand, we have different receptors. And some of the activated early in the some of the activated at the low concentration, some activated at the high concentration. That basically is a feature of the biophysics of the receptor-ligand interaction. If the connection is strong, if the molecule very well fit to the pocket, it will be activated in the low concentration. If it's not well fit, it will be activated at high concentration. So when we talk about ligand versus non-ligand for a given receptor, you need to think about the concentration because many receptors got activated by orders at a high concentration. Now, when you present an order at a given concentration, you activate some number of receptors. And that's exactly what you see. You activate 2 receptors for the order of the small concentration. You activate 6 receptors for that order, for this concentration, and 4 and 6 for high and high. So as you increase concentration, you recruit more and more receptors. Now look at this picture. And now what feature of this pattern is irrelevant for the concentration? Obviously, and well, the intensity may be different, and intensity is actually difficult. I mean, first of all, it's difficult to measure. Second, let's for now a little bit forget intensity and discuss it later. But look at this pattern. What is an invariant feature that is common for all 3 patterns? Obviously, these 2 receptors. These 2 receptors are always activated independently of the concentration. Okay? Now, but if you're looking on this without, how do we identify these receptors, for example, in this pattern, if you present this concentration? You present this concentration, how would you choose not that 2, but these 2? And the idea came to our mind that this is actually temporal sequence of receptor activation. So the receptor that activated low concentration activated early in the SNF cycle. HF, HFAG, HFAG-LK. So how about we'll pay attention for the early activated receptors. Tick, tick, primacy coding. This is the most relevant. That's exactly what we talked about it in the previous experiment. The early activated receptors are more relevant for odor recognition. They're telling you more information about odor identity than the rest of the code. Well, that's what we call primacy coding. And let's just make a very simple model that to identify the odor, we need a P number of odor receptors to be active. As soon as you activate 2 receptors, you know that this is apple or this is coffee. Or maybe it's a timing. Now, that is actually dependent on the specific neuronal mechanism, and we can discuss a little bit later. So the primacy window is defined either by the number or by the timing. So it's a concentration invariant by construction. So we build the code, we build the concentration invariance in the code. We need to discuss what are neural mechanisms for building this code, how the code can be read, what are the basic coding features, and is it actually with orders is what we observe? Because we observe it with patterns. Can we do experiment to prove the relevant window of information for the order patterns? Let's talk through this a little bit. So first of all, how we can build the code. I mentioned this a little bit previously, that when animal inhaling the order, it's inhalation, then pause and exhalation. So the concentration in the nose goes slowly and more or less correspond time scale with the time scale of the inhalation. Okay, it's increasing when you increase the when you're inhaling, you bring the molecules in, and as the concentration crosses specific threshold, you activate a specific receptor based on its affinity, and the most sensitive activated earlier, less sensitive activated later. So you have a sequence of receptor activation, and basically this axis is affinity. Affinity. So the higher affinity, the earlier activated receptors. Okay. Now, and that is defined by the concentration of the external stimulus. Now, what happens if you increase this concentration of the external stimulus? You basically making the slope steeper. You basically scale up this curve, and a receptor will be activated earlier, and you activate another receptor that hasn't been activated by the low concentration, what we observed both on imaging studies and in kind of schematic models. So what is crucial here, the timing shift earlier, and we activate more receptors. The idea that the order can be detected, order identity is set up by sequence of receptor activation has been proposed by Hopfield. You need to know this work, 1995, and was dominant in the field for a while. Basically, the timing tells you the concentration, but the sequence tells you the identity. And this is two independent parameters that is useful to observe. And that has been observed. So if you increase concentration, you see this timing changes here. What we actually proposing, and later work, not only ours, but other work, propose that it's not just only timing, but the early is more important. Whole sequence doesn't matter. And that is actually an important part. You remember the idea with the tennis player? You have a correlation with the whole trajectory, but early is important. That's the important part. So how this code can be read? You have this activation of the glomerulus, HF, AG, LK, and they activate a bunch of mitral cells. And mitral cells that activated in some temporal window converge to the piriform cortex. This converges to this cell, this converges to this cell. So imagine we have here the inhibitory network. Inhibitory network. Then those who activated earlier set up the stage and suppress the rest. Oops, sorry. Sorry about that. Those who activated early through the inhibition suppress the rest. This is an interesting idea because basically then you're paying attention only on the early activation and the inhibition suppress the rest. But what happened, for example, if you increase concentration significantly, so you squeeze more glomeruli in the first temporal window, then you activate more cells in the piriform cortex. And that actually phenomena that is known that order, if you dramatically change the concentration, change the identity. Here's an example. Amyl acetate is an order that smells like banana at a low concentration and like a nail polish at a high concentration. Now, if you take a nail polish and just slightly puff in your nose, you actually will smell banana, but not a strong one. Okay. Recent work by Kevin Frank's lab demonstrated that while the olfactory bulb has a lot of activation during the whole 300 milliseconds, piriform cortex is activated early and then suppressed, and this is pattern is concentration stable, concentration invariant. Concentration invariant is a strong word. Maybe concentration stable, concentration tolerant. Invariant is too strong word. It is varied, but there's a feature that is invariant to the concentration, changes from the concentration. Now, there's another interesting ideas about the code. Let's assume that you stimulate glomerulus CATMW in this time sequence. So this is order cat. And you present a different order. This is order dog. Now, what happens when we mix cats and dogs? Actually, the first three become important. That became a new object. It becoming CAD, CAD, with C will be most important, D is likely, but that's a signature of a new order. Now, you remember, if you change concentration of individual components, you shift their timing. So if you decrease the number of cats and increase the number of dogs, you change the concentration, and this is a different object. This is become COD versus CAD. And now we can start building the different mixtures. And that's actually, if we would be paying attention to the whole sequence, it will be much easier for us to identify to extract dogs or cats from the dog and cats mixtures. Actually, when we smell coffee, you don't know what components in the coffee, because in your head, it is basically that's the object that is created in your head. And again, this is due to the primacy effect. Now, when I say other concentration invariants, what do I mean? Basically, I know that it intuitively obvious for us that order is perceived independent of concentration. How should we check it for animals? Can we design experiment that prove that animal can detect orders independent of concentration? And when I talk about orders, I actually mean the order is actually coffee is not constituted. So when you change concentration of the coffee, bring it closer or further away, you proportionally change concentration of all ingredients. It's not that you bring one ingredient high, another low, then you may mix up the coffee. But if you make them proportional, you keep the invariance. So how can we check it with the animals? Beautiful experiment has been done by Zach Manuel group and now Uchida is the following. So this is quite important. This is a concentration of order A. This is concentration of order B. And let me draw this picture here. So this is mostly, this is A. This is mixture of A. This is pure A. This is pure B. And along this axis, this is binary mixtures, okay? And we can train an animal to discriminate these mixtures. So the more A-related animal go to the left, more B-related animal go to the right. And the decision boundary will be this, okay? This decision boundary. And that's exactly what they did. So here the data. So these four mixtures go to the left, this will go to the right, okay? But then in the probe trials, they change the overall concentration. They decrease it twice lower. and ask, for example, here, where they go. If they measure the ratio, then the decision line should be this. If they measure, for example, one of the components, the decision line should be that. So, but what they saw in the result, that the probe trials fully match the previous one, which tells us that the decision line is concentration invariant. So they did it proportionally to initial concentrations. They did exactly the same thing by changing concentration not 2 times, but 10 times, which is becoming much noisy, and also by increasing concentration, which became almost the same. So this is a hard experiment to do, guys. Behavior with animals and interpretation of behaviors is well non-trivial. For example, we tried this experiment. It's not easy to kind of work it out, but it's a beautiful result. So in some range of concentration, here basically it's more or less tenfold. From here to here, it's tenfold, and tenfold down and 2-folds up. Animal perceive odor mixture independently of the concentration. Okay, and that is actually very well tuned to the idea that, you know, if you activate animal paying attention to the early glomeruli. Now, what is the capacity of this code? Imagine we have n glomeruli, and P is the number of primary glomeruli that we need to pay attention. Okay? Then the number of potential combination will be n factorial divided by n minus p factorial p factorial. If p, let me change the color, sorry. If p is much smaller than n, it can be n to the power of p divided by p factorial. Because basically n divided by n minus p, you can kind of neglect n minus p. You can basically just take only the first n components, approximately this. So for us, is n equal 350, p equal, let's say, and I don't know what p is, but if p equals 6, then total number became 2 to the power of 10 to the 12. 2 trillion odors. Huge capacity. So paying attention not for the whole code. The whole code capacity will be 2 to the power of n, binary code. But paying attention also on the binary only on the first p, give you huge capacity. Do we have this capacity? We don't know. But there's a humongous information capacity of the code. So this is the answer to one of the previous questions. By the way, here I would like to make some small deviation. There was a paper in 2016, 17, I don't remember. Humans can discriminate trillion odors. Guys, this paper has I was super excited. When I saw this paper, I immediately plugged it in and get this number. This is trillion odors. I thought, oh, now I suddenly know the P number by measuring the information capacity of the code from different experiments. This paper has a very significant flaw. And I can discuss it, I don't want, but basically not kind of, they didn't take into account very important parameter. There was a lot of discussion in the field, but uncertainty of their discussion was 10 to the power of 12 plus minus, say, 9. So take this paper with a grain of salt. Be very careful about this paper. But we don't know the capacity of the code. We don't know the real capacity, but definitely the primacy model would not limit us, because even p equal to 10 will be huge number. I'm not sure that humans have this resolution.
Dima, there's a question.
Yes, until now, the components of orders have been temporally separated, and the effect of concentration to primacy, etc., have been interpreted. But could it be possible that components of two different order activated two different glomeruli simultaneously? What happens then? Could there be additive inhibitive effects on the activity of the glomeruli due to the simultaneous activation of ones? Absolutely. So first of all, simultaneity is a difficult concept because it's what you call by simultaneously. If they activate it in 20 millisecond time window, maybe it's already simultaneous. You need to think of the yes, exactly. Thank you very much for sending this paper. Sorry for this for distraction. So I just saw this link, immediately clicked on it. Thank you, Banu. So coming back to the Shiva's question, it's not clear what is simultaneity here. Basically, when is this code is different from there, or from if this is code is different from that code. I mean, yes, it is, and it's all we need to dig into the neuronal processing and how it's going on. And behavioral experiments are very slow because changing anything requires hundreds and hundreds of behavioral trials and noise statistics and on. So we're limited. So take it as the hypothesis generation mechanism that you need to dig and to think and write models and do next experiments. Because testing your ideas may take us a couple of months just to change the timing of the patterns simulation. So basically, some of the we don't know. Some we can check, and it's still in the process of discovery. So, okay, now, can we actually prove with orders that the temporal window is important? And not with a pattern simulation. And that's what we explained that we did. I have a few minutes, and that will be good final. I have about 10 minutes final story. We basically train an animal to discriminate order A versus order B. But again, to make the task more realistic, we scramble the concentration to order of magnitude. We presented, and independently of the concentration, animal need to go left or right if it's order A or order B. An animal can do this task very well. We did it very precisely. We measured the pattern of how order activated with so-called photoionization detector. We know exactly when order comes to the nose. We trigger the order valve. When mouse, we measure the sniff. When mouse starts exhaling, we trigger the order valve. So this is when mouse started exhaling, this is the beginning of the sniff cycle. That point is super important. Sorry, just let me draw again. This point is very important. This is the time when the mouse started inhaling. And in some number of trials, we perturb the pattern by presenting a very powerful light pulses in the with optogenetically tagged O receptors. We basically blast the light into the epithelium, and O receptor has channelrhodopsin. The idea of this experiment is the following: imagine the imaging task. Do you see me? Actually, so I give you one pattern versus another pattern, but I show you it very briefly. But in vision, vision experiment, you can remove the pattern very quickly. But I cannot remove the pattern once I showed you because order has some dynamics. But instead of removing the visual pattern, I blind you with a huge projector into your eyes. I basically have on the side a huge projector. I show the pattern and then blast the light. And I do this with a specific time window after order onset, which I consider to be the onset of the inhalation. Again, show one pattern, wait a little bit and blast the light. And I do it on a very small subset of trials on intermediate concentration. Again, doing behavior, you need to think very carefully what animal is extracting, how to interpret the experiment. We train animal on that task, and animal learn the rule. The rule need to be learned. A go left, B go right. And the mask should not be informative, should be very rare, and animals should not learn from the mask. So we did the mask on a small number of trials. And that more or less corresponds to the following. The normal development of the patterns is, we present an order in activate glandular 1, 2, 3, 4, 5, 6. Versus something like that, 1 and then a blast, or 1, 2, blast, or 1, 2, 3, 4, 5, and then blast. So how many glomeruli, what are the time we do? We can't count really glomeruli yet. How many, what are the length of the time we do that is important for the order discrimination? Without mask, at both concentration, high and low, this and that, animal perform almost above 90%. Okay? And now when we present the mask at different timing in the sniff cycle, performance was like that. So initially, performance was about 60%, and then it increased and goes to the almost to the saturated level. And that was at the low concentration. What is the prediction? What will happen with this curve at the higher concentration? High concentration should bring orders earlier. So the mask should be short, and that's exactly what we saw. The mask shift here. What's important here? This is, first of all, the temporal shift of the mask more or less proportional to the temporal shift that is evoked by this by shifting concentration in the neuronal dynamic. I don't give you data on the neuronal dynamics, but it's in the same order of magnitude. You shift for 1 order of concentration, you shift the neural response 10 milliseconds approximately. The second thing, what is important, that, that this is an important temporal window. That's where the information is accumulated. And this is approximately in the first 100 milliseconds. After 100 milliseconds, this part is irrelevant. Now, where are we standing for that? Where are we standing for the order identification? You remember this experiment when Vijaykumar and the first author Kerry measured the activation of the glomeruli with calcium imaging as a function of the time latency. You see this distribution. There's many orders, many glomeruli. This is distribution. Somebody asked me a question about the shape of this curve. But basically, some glomeruli activated very early. This is probably 0, it's on the level of the noise. But some activated 300 milliseconds. Where we are with the accumulation of the evidence in our task. We are here. So we need this portion of the glomeruli to make a decision if it's order A or order B, which is proportionally, approximately 10% of total number of glomeruli. This is a distribution for all orders and all glomeruli. But if we scale down for one order, so in one order activate approximately 100 glomeruli at high concentration at a later time, so we need approximately 10 glomeruli to identify the order. This is very crude estimation, but that gives you some ideas what may happen. So animal need very few glomeruli to make a judgment for order identity. Why do you need the rest? Very good question. What's happening on the mitral cells? You remember this curve. And my statement is that that's a portion that is very relevant for order identity. And that portion is a part of the trajectory that the tennis player do after the ball leaves the racket. Why do we need it? We may need it, we may not need it. I don't know. Maybe this inertia in the bulb, you kick the bulb and bulb will do something, so inhibited, disinhibited, and continue. You need to finish the swing. You cannot stop here. You cannot be completely efficient, not do any extra spike. You hit the ball and stop the racket. You cannot do it. You have inertia. So maybe some features of the code are not defined by information transmission. from the behavioral perspective, the most earlier the important, the later we don't know the origin. Or maybe animal is using this for something else. I just give you some example, just provocative example. I don't know how to test it, but we need to think about it. Imagine I train you to swing the racket. What will I tell you? I wouldn't tell you stop when you hit the ball. I said, move your arm to the left or move your arm to the right. Maybe this is a learning signal because it's very well correlated with the stimulus. Maybe animal extracts some information when it learns the order. We don't know. But for pure coding perspective, that's what we established. We established that the earlier part of the sniff cycle, earlier spikes are relevant, the later are not. And that has really relevance for the order detection. Orders can be identified in concentration-invariant manner by the earlier glomeruli. What the system is doing later, we don't know. Let me just briefly conclude this portion. The primacy coding model is concentration-invariant. We talked a little bit about mechanism for forming the code, mechanism for reading the code. The code is consistent with some known behavioral phenomena, and a small temporal window is relevant for behavior. And the summary for today's lecture is, again, think always in this way. Stimulus neuronal behavior, think a lot about this arrow. We can extract a lot of no kind of understanding by trying to understand this part. And it's not trivial to do this experiment, but so far we can ask the question about which features of the code are perceptually accessible. An analogy, for example, we can sense a single photon with a retina. This is perceptually accessible. Do we use this feature when we recognize the faces? Probably not. It's a very different computation, but that sets up the limits of performance of your sensory system. So now we can study this with the inside of the code, inside of the brain. Or we kind of develop the paradigm for evaluation of behavioral relevance for coding features. It's very artificial, but it gives us some ideas. And we think how to link it to the real orders, have the model of this code. Now, when we have the model, by the way, we can actually start comparing with the real orders. And we propose primacy coding for concentration invariant, and basically earliest part of the sniff cycle more important for order identification. Questions?
The question.
Cortex, okay, sorry. Can an animal distinguish between two patterns of genetic activation in which the first P are identical, but P plus one is activation is different? Yes, it can. Beautiful. So initially, and I can talk about it a lot. So basically, this is a beautiful difference between what is detectable and what is used. You can train an animal to pay attention to single glomerulus activation late in the sniff cycle. Here's an analogy. You can train yourself to use your peripheral vision to judge the number of fingers I show you, 1 or 2 in the periphery. You can do it. But if I ask you how many fingers you foveate, you turn your head, you turn your eyes, and you make the judgment. Yes, you can train an animal, and animal can discriminate features of the code later in the sniff cycle. Period. No question asked. We did this experiment, it can be done. In an object recognition state, when you ask us to recognize a face, how you do it, that's a different question. And that is actually much harder to assess. And that's what we have been studying. Okay, hold a second. Okay, was it clear? I hope it was. So the answer to Sharad's question, yes, it can. Banu, question. Hanu question. Do peripheral cortex recordings support the primacy models as well? Yes, indeed. Let me try to, oops, hold a second. That's exactly the picture that I showed you, that, that peripheral cortical cells are, have the strong spike that is concentration invariant and early in the SNF cycle, and they basically respond only to the early activation of the bulb, but not later. And what's interesting, when you suppress recurrent connection by pharmacology, you lost this feature. Beautiful work, 2018 in Science by Frank's lab. Other experiments comparing behavior of younger and older species. I'm thinking the younger ones utilize the later part of the plot, but then they deem automatically unnecessary by the older ones. Potentially, yes. We don't know. This is a hard experiment to do. And studying these age dependencies, because you change many parameters simultaneously. We don't know yet the basics. My take on it, let's think about the general model of the code, and then we see how this model changed with the time. We're still in the process of building this model. Actually, for example, what I really would like to do, I would like to do these experiments that we did with optogenetics with others. And what we're now developing, because it's all based on the fact that animals need to judge perceptual distances, how similar order A to order B, which is different from perceptual discrimination. Because it's easy to train animal to discriminate two orders, but then you don't know what feature of the code is extracting and how it is related to natural behavior. And this is a hard experiment, and we're developing some methods to make animals judge perceptual distances, and then we perturb in the code and seeing how these perceptual distances are different. What I think by perceptual distances, I mentioned this before, then if you intuitively know that orange and apple are far away, much further away than orange and lemon. You have this notion, it's somehow formed. Can we perturb these distances? Can we measure these distances? Can we make animals to report these distances? This is a harder problem. Look at our bioarchive paper in my website. We're now trying to make this measure, but it's the work in progress. So we're not yet studying the young versus adult. By the way, here's my take on it. The primacy window is not fixed. It's not that we fix the primacy window. It's basically, let's imagine you train the wine, and maybe perfumer has a much longer primacy window. They extract much more information than we have. That can be trained, that can be the subject of attention. When you taste the wine, the first thing, oh, this is red wine. This is a Cabernet Sauvignon. This is 1997. This is from Bordeaux. So you build up more and more detailed description. But early decision is important. This is why. This is getting the first few tenths of millisecond. Okay. Could it be that it's not perceived order, but the expectation that of what is perceived is encoded here? By here, what do you mean? Sharam? I'm reading Sharam Narayanan question. Could it be that it's not perceived order, but the expectation of what is perceived is encoded here? You meaning at the end of the SNF cycle?
Hi. I mean to say that, so the mice are trying to make a behavioral decision as fast as they can, because that's probably advantageous ethologically for them to do so.
Yes.
So the faster they do so, the better it is. But in cases where they are incorrect, I mean, like the previous question where you said if the first P glomeruli are the same, they can still distinguish. So it could be that here they are just completing the pattern because they expect the odor to be something rather than this being an indication of odor perception itself.
So, okay, does expectation play its role? Yes, it does, for sure. And it's actually super important. And your perception, your expectation, specifically for odor processing, is super, super important. So a lot of basic experiments have been done in food sciences. When the wine connoisseurs here was asked to judge wine, and they changed the color of the wine, they were giving wrong descriptors. They were describing wine in a very different way, because you expect something and you get something different. It's a known fact that your perception of the odor is very much based on the expectation. Absolutely. In this experiment, I don't know. It's still, I think that early part is more important than later. Does it affected by the expectation? Yes, but I can speculate. I can actually give you some ideas. For example, maybe you remember that glomerula goes to the mitral cells. Mitral cells has a granular cells connection to the granular cells, and this is different glomeruli, and this is. But granular cells have connection from the cortex. Maybe your expectation suppresses these mitral cells and make these mitral cells earlier. I don't know. This is just found hypothesis. That maybe your feedback messing up with primacy. I don't know how to, we're thinking how to test it, but that's kind of consistent with the primacy coding idea. So your expectation sets up the primacy. In your case, the signal initially, it is different if you go target versus non-target odor. So animal need to make a decision. So it's not pure expectation signal, but expectation does play a role. I'm very open for discussion of different experimental setups and how to test it and what's actually coming out of this experiment. But that's, guys, this is the cutting edge. We don't know. This is a fantastic field, which is less explored and very kind of prime for this exploration. A very good question about expectation.
Okay, they don't see any more. Thank you, Dima, for a wonderful set of talks. We should all thank both Dima and Antonio for the very nice set of talks they've given over the past 2 days.